but he got his PhD from the University of Otago, which I'm assuming is in New Zealand.
Yep, it is.
And then did postdocs in Kent and Oxford before returning back to New Zealand, where he's been studying genomes and cell structure and really looking at the relationship between genotype to phenotype, and using those relationships to understand not only disease but biology in general. I hope that's an adequate description of what you do. He gave a wonderful presentation actually at Penn State physically here in State College, maybe 6 weeks ago, something like that. So he was here, and he talked about something not microbiome related, and I'm really excited to hear about his microbiome work now. So everybody virtually clap for Dr. O'Sullivan. Here we go. Thank you.
Thanks very much, and thanks for coming. I haven't given one of these this way before, so this is kind of a new experience for me. It's also about 4 o'clock in the morning, so I'm just waking up a little bit. So I'm going to talk to you now about the microbiome work, and so obviously, I mean, you guys are all microbiome people. I just got to shift my bar here. But there's clearly, there's more to us than you see, and that's true, of course, with everything we do. It's true in terms of microbiome, but it's true in terms of the work that I'll show you as well. So the work I'm going to show you today is done by a large group of people. So a lot of it's being led by myself and Wayne Cutfield here. Wayne's a clinical endocrinologist. And so we've led a lot of the studies that I'll show you today together. And then the work on the preterm infants that I'll show you was largely done by Thelini and Valentina here, with some help from Cam and from Sash in here. And Tommy has worked, of course, who talked a couple of weeks ago. He's helped with analyses, Liz with some other stuff, Jose as well. Quite a lot, and then a large list of other people. And it was funded by quite a wide range of people. The National Centre for Growth and Development, which is a centre of research excellence in New Zealand, are virtual centres that are funded by the government. And then some of the SB and paediatric endocrine societies, etc. So you all know that we're a tale of many genomes, and that the microbiome that we have on and in us effectively is a plastic genome that can change its structure in response to environmental signals and developmental stages of our life. And so it's a part of our genome that can change effectively, alter and give us parts of the things that we require and as we go and as we grow and as we age, etc. And there are a large number of common features in the microbiome. And there are common features as it develops. At least it seems that there are common features as it develops. Now, we've got a number of studies, of course, that are being done currently that are looking at this. We're doing studies and other people as well, right from preconception through birth and into early ages. And so there's one study we're running at the moment called the NIPA study, which is looking at that going through preconception, which is actually really interesting. But this one here shows basically some of the patterns that are thought to happen, the general patterns that are thought to happen as children age from birth through to 3 years. And so there's a period of life here where effectively we have quite a lot of change. Let me just see if this should work. So this part here, sort of between 6 months and 24, is thought to be quite a dynamic phase of life with your microbiome. Changing quite a lot. Levels of Bacteroideaceae, Lactospiraceae, etc., all shifting around and changing relative to each other. Here, of course, it's thought to be very sensitive, and it's sensitive to the birth mode and to your predominant nutrition and to antibiotic use and other factors, environmental factors and hereditary factors from your parents. After that, of course, it moves as we transition through the solids into some sort of more high diversity and resilient to change sort of model. And then that's thought to stay with us for a long time. And that's kind of what's thought to go on. We know that depending on how you look at this, and of course numbers in the studies are changing now and things are stabilizing out in terms of what we understand, but that sort of the birth mode and then the way you're fed, etc., all seem to alter the microbiome at the different stages of your life. And so on the basis of this, we kind of wondered a little bit about what happens longer term. And so preterm birth is very common and is associated with significant stress and metabolic change. And so preterm birth is anything where you're born before 37 weeks. So there are different stages of preterm birth. Between 34 and 37 weeks is generally considered to be moderate or late preterm birth. So the child is reasonably well developed at that point. But of course, children can be born and can be, I want to say grown, because that's basically what happens. They're born at 24 weeks and they're effectively grown in an incubator until they're of age and capable to basically look after themselves. Live without intervention. But being born early like that is associated with significant stress. There is huge physiological stress on the child. You're under different forms of nutrition. So you're doing parenteral nutrition, which is direct into the bloodstream. There's nothing through the mouth. If they're born particularly early, they can't even suckle. You have dietary fortification as a result of this. So you're not getting what you would normally get through breast milk. In fact, the parenteral feeds that people are given are actually have been developed in a very hit and miss way. So the composition of them is very unusual and differs between sites even. You get a large number of infections and antibiotics treatments as a result of that, and to prevent infections. And of course, you get given steroids and things. I mean, the steroid treatment is one of the first things that happens normally to the mother. And the reason for the steroid treatment, of course, is to inflate the lungs, or to enable the lungs to inflate. In the absence of that, it's one of the greatest interventions that was done really around preterm birth, because in the absence of that steroid treatment, the lungs don't inflate. The child is born and the child can't actually breathe, and they used to die. But with the steroid treatment, of course, they come out, their lungs, the water tension between the surfaces and the lungs is broken and by the surfactants, and so effectively then the lungs inflate and the children and can breathe. So as a result of being born preterm, and particularly anywhere around 24 weeks through to 32, in this period where you're born very preterm or extremely preterm, the treatments are pretty extreme. Children are put in incubators, humidified constantly. Very early children can be wrapped in plastic to try and retain water within the skin and things, and they are treated in a very particular way. So, this is a very significant stress on an individual. We know that preterm children who are born this way have later life complications. And those later life complications include things like increased rates of obesity, diabetes, metabolic syndrome, but there are also some individuals, depending on how very early you're born, who show significant learning capability differences, and a whole lot of things that happen to them. But it's really the things around obesity and things that we were interested in, and diabetes, because if preterm children are predisposed to the development of that, then why is that? There's nothing different in them with respect to their genetics, really, at least as we understand it. But is there something else? And so, of course, the microbiome is an obvious thing to look at. So our hypothesis was that there are alterations to the microbiome in preterm children. And that's kind of simple to think about from birth. At birth you could look and you could say, well, the microbiome is going to be different. But the question is not whether the microbiome is different at birth, because there is a sort of understanding now that as you go through birth, the microbiome, from birth to early stages of life, the microbiome will come towards a sort of a more general biome structure. And if that's the case, then hair would really matter. or are there long-term changes in the microbiome that we see with preterm infants? And so with respect to that, we were asking a question about what happens to their microbiomes when they're out here. So when they're about sort of 8 years of age, are their microbiomes still significantly different, or are they the same as they were, or are they different? Are they still significantly different, or have they come towards a consensus with the normal birth children in the environment that they're in? So we designed a study, and what we did is we retrospectively collected a group of people, a cohort of children who were born very preterm, so between or before 32 weeks, and extremely preterm before 28 weeks. So they went from 24 through to 32 weeks when these children were born. That's 24 weeks of gestation. So very, very small individuals when they were born. Term controls were recruited, and they were between 37 and 40 weeks of age. So a standard sort of gestation. And we recruited these children when they were between or at about 8 years of age. And so they were slightly different in age. The preterm children were slightly younger on average than our term controls. But we recruited these children, 50 in each group, from the population here in Auckland. We then tested them. We ran insulin sensitivity tests on these children. We did 24 ambulatory blood, 24-hour blood pressures on them. We did body compositions, so that's a DEXA, so it's a whole body X-ray scans. We did metabolic syndrome markers on these kids, 3-day food diaries to see if they were eating differently, because there is a thought that preterm children can be treated slightly differently by their parents, so very precious, and some of them get given particular diets and things. Microbiome on them was tested, and the metabolome was tested as well. Okay. So the cohort, the inclusion criteria that we had were that they had to be healthy and prepubertal. We didn't want to get into the situation where we had some children going through puberty and through the menarche. So they were healthy prepubertal children. And they were excluded if they had any evidence of gestational diabetes or hypertension, if they were conceived by IVF, if they were small for gestational age. So that's not relative to the other group, of course, because preterm children are very small. But if they were part of a multiple birth, if they had any necrotizing enterocolitis, siblings in the study, or first degree relatives with type 2 diabetes. Okay. So the dietary intakes between the groups, and here we have, this was done on a subset, so it's on 20 and 20, well 18 and 18, sorry. But here we tested this down, and the dietary intakes on these individuals was broken down completely from a 3-day food diet, and the dietary intakes were not significantly different. And so that's kind of cool. So we were pretty happy with that. When we run through the demography of the group, what we saw was that the gestational age, of course, was dramatically different, which is, of course, a selection of the group, which is good. But other characteristics in terms of their demography, their age, the sex ratio, the ethnicities, those weren't different between the groups. Ethnicity was moving a little bit towards, trending towards a significant change. And that's because some populations here do have differences in their birth rates and preterm rates. In terms of their birth weight standard scores, standard deviation scores, again, they weren't different. Delivery by C-section was different. Some of the preterms born more frequently than by C-section than the others. That's obviously for emergency reasons. Other characteristics though weren't. In terms of their childhood anthropometry though, so this is when we had them actually in the study at 8 years of age, that was slightly different. So the weight SDS was different. The preterm children were slightly heavier. The height was different. And the BMI also was different between the two groups. And the preterm children were basically showing evidence of being pre-diabetic. We then looked at a number of factors in these individuals. So fecal calprotectin was measured from stool samples from the individuals. We took stools at a fasting time point from these kids. And the stools basically were analysed for a number of things, and fecal calprotectin was one of them. So fecal calprotectin is a marker of inflammation in the gastrointestinal wall. And so what we saw here was a significant difference in the preterms. Now, none of these children were recognised as having any ulcerative colitis or IB, inflammatory bowel disorders. But the fecal calprotectin levels in these kids, particularly in the preterm children, were very, actually got to be quite high. So there were a number of children in the preterms who were actually up around the 1,000 milligrams per kg. And those kids are getting up to the point where actually they were showing symptoms, and they were recommended to be seen for IB, inflammatory bowel disorder, and be checked up. But what we did see here is that there's a large number, of course, of individuals who are down around the baseline of the detection for the assay, which is what you expect to see in children. But then the children who were preterm show a greater distribution spread up than the children who are term. So many more term down here, and what you see basically is a significant difference in terms of fecal calprotectin levels in these children, which indicates that the preterm kids are showing signs of inflammatory or inflammation in their bowels. And given the fact that they're living in the same environment, the socioeconomic groups weren't different, that they are having effectively the same diet, then this is a really interesting finding. And it may indicate that there's a change in terms of the microbiome. So the question then, of course, is, is the gut microbiome composition different? And as you guys are all aware, there are multiple ways to do this. You can use your 16S amplicon sequencing, and you can basically look at the list of who's there, or you can do your metagenomics and look at a functional capacity that's present in a metatranscriptome, and look at the functional capacity that's actually being expressed in the children. And you can do these different ways. So in this study, we did all of them. We did the 16S amplicon sequencing, we did the metagenomics, and we did the metatranscriptome. We did 20 million reads for the metagenomes on these kids, and we did about 20 million reads for the metatranscriptomes as well. The metatranscriptomes looked really great, but actually they failed, and we're not quite sure why. But what happened was that there seemed to be an overabundance of particular transcripts throughout the population. And when we collected the fecal material for the metatranscriptome, we collected using RNA later. And the samples were collected into an RNA later solution. And for some reason, we think that something went wrong with that. And there were hydrolases that were expressed across the population. It was very, very unusual. And so that data, unfortunately, didn't show anything. We have subsequently done other studies where we've used the RNA later using the same protocol, because we'd already started those, and those ones were fine. So we don't really fully understand what happened with the metatranscriptome data. But both the 16S data and the metagenome data worked really, really nicely. And so the metagenome data is particularly what we focus on more. So the bacterial component of the microbiome did not differ in children born preterm or term. And it didn't differ in children born preterm or term by either method that we looked at it. So that was both the 16S amplicon sequencing and by the metagenome. If you look at the core microbiome and then the minor parts of the microbiome, they were effectively the same. There was a very minor change in the core microbiome between the preterm and the terms at 8 years of age, but it was nothing significant. And this is really unusual because we thought that given the fact that we had seen the sort of difference in calprotectin, that maybe there would be something different in the microbiome. and it didn't really make a hell of a lot of sense. But what Tommy did was he actually looked at the phage component in these individuals. And so we looked at the phage richness, and we looked at the differences between the children who were born preterm and the children who were born term. And so bearing in mind that these kids are now 8 years of age, been living effectively in the same community, getting the same, exposure to sanitized water and everything else, we thought, well, that these things would come together. But actually what we saw here was that the partial phage genomes per million genes, so this is how we standardized this one, for the preterm children, they were a lot lower and significantly lower than what we saw for the terms. And that was if we did it by what we've got here on the left is the partial phage genomes per million genes. You see here that the preterms are running at around about 450 odd as an average, whereas the terms are running at about 550 odd. And you see here that the partial phage genomes per 1,000 assembled contigs were the same, okay, showing a difference again, a significant difference between the preterms and terms. And so this is quite interesting. Because this means that even after all this time, that the phage components in the microbiomes between these two groups are different, even though the actual component microbiomes are not, in terms of the bacterial components, etc., that we saw. So we looked at this and we tried to understand a little bit more about the phage and what they sort of were and what they represented. And of course, there are ways to do this. And so we ran the correct programs and we started looking at them. But the problem with phage is that most of them are unknown. And so you can do this, you can get your nice network maps of them. And you see that, in fact, they are largely ones that, within this map, are largely ones that are recognized as being associated with people. But the identities of the phage are actually really poorly understood. And so this kind of didn't help us a lot at all. But we do know that there are significant differences, and we are continuing to try to work in this, and Tommy is starting to take this off and run this on his own as part of other studies that we've done as well. So it's a really interesting area. We know there are differences, but we still just don't know enough about the phage to really say what they are. And that's a bit of a shame at the moment.
I have a question. Thank you. I don't know if it's better to wait until the end or now.
No, ask now, it's fine.
I'm very interested in like viral populations and especially phage and their interaction with bacterial populations. So did you study any relations between the populations of certain type of phages and certain kind of bacteria in the gut of the human or something? So, yeah. And also the second question is, yeah, most of the phages are unknown and this is a problem. So how were you able to enrich for phage populations or after you got the sequence data?
So how were we able to arrange for what, sorry, phage?
Enrich, yeah, the phage populations in the reeds.
So, I mean, this is a very good question. In this study, we did not go back and look at the phage identities, or at least the genomes that we were able to build. We didn't look back at the bacteria that they were associated with in terms of trying to correlate those together. The problem that we had was that the phage numbers were different between the two populations, and yet the microbiomes didn't show any difference in terms of alpha diversity or beta diversity. So we were kind of stuck with this, it seems kind of to be a really weird thing, right? It's based on the metagenome data. The one part comes out as being different, which basically the predators of the population are different, and yet the population itself doesn't show any significant difference, which is a weird thing to kind of come back to. With other studies that we're doing now, we're increasing the coverage we're trying to increase the coverage and trying to get better so that we can hopefully reassemble the genomes more completely and then have phage actually inserted where they are inserted into bacterial genomes, at least for those ones that go into the sort of lysogenic lifestyles and temperate phage. But in this study, we weren't able to really do anything more about that. It was we weren't expecting to see the difference in phage, and so we can go back and do more sequencing on this, but we kind of have left this one alone as where it stopped. We were thinking about why this would occur and why the phage would be different in a preterm population like this. And one of the things that we kind of thought was that it's about the way, the hypothesis we came up with and we presented, I think, in the paper which is under review, was that we thought it was about the way the phage were populating or colonizing the individual. So one theory about the bacteriophage is that the bacteriophage are there and are entering into the microbiome, and they're there in individuals very early in life, and what they're doing is preventing colonization by bacteria that are pathogenic or shouldn't be in the microbiome at a particular stage of your life. And that what then subsequently happens is that the phage population, as the microbiome develops, it's there and it's knocking out anything that's coming in. But then as that happens, the phage population starts to crash. And so if there are no hosts for the phage population, then the numbers of those phage will crash and they come down and down and down and down. And so the diversity in that population reduces. And what we kind of argue, at least in the paper, is that what's happening with the preterms is because they're getting nil by mouth for quite a long period of their life, particularly these ones who were born at 24 weeks through to 32 weeks, they are, excuse me, getting nil by mouth. So their gut colonization isn't occurring, and so they're not getting the hosts for the phage. So while the phage are there, the population is dropping because there are no hosts. And then it's coming down until the point where the hosts arrive, and then the population and the diversity in that population is crashed and is held much lower level, and it stabilizes at that level. I mean, that's only a hypothesis, and we don't know whether or not that's right. In fact, we're not even really sure how to monitor that one fully and to test the hypothesis fully, but that's at least kind of what we thought might be going on, or what we proposed. There were a number of other changes that we saw in these children. We saw differences in the fecal levels of a number of different compounds, butanoic acid and pentanoic acid as well. They were higher in children born preterm. The fecal levels of the methyl isobutyl ketone were also higher in the children born at term than they were in the ones at preterm. And there were a number of other plasma volatiles as well that were different between the groups. It was about 35. And so this is really interesting because, again, we're thinking, well, the diets should be about the same. Well, we're the same as far as we assess them, but there are some products here that are of bacterial origin. They are different in both the feces and in the actual plasma of the host as well, of the children. Amino acids were one other group where we saw differences. We saw some differences in terms of things like arginine, which of course is a non-essential amino acid, or conditionally essential, amino acid. And its levels differed again between preterms and terms in the plasma, but not in the feces. And so these things, they indicated there definitely were differences here. And the argument that we had there is that basically they're representing this inflammation of the gut is affecting the way that they're transported into I keep saying the host, but into the individual from the gastrointestinal tract. There is also the possibility that some of those amino acid changes that we saw, because we didn't look at this dextro-levo or the RS so the enantiomer ratios of the different amino acids. We are currently looking at that because we think there may be differences in terms of ones that were actually L or D, or cell bacterial origin or not. So collectively, we thought that these observations are consistent with preterm birth being associated with long-term impacts on the microbiome. The bacterial component at least seems to stabilize out and seems to come towards a consensus, but the phage component is different and does represent a significant difference in terms of the microbiome. Unfortunately, we don't know enough about the phage, and so it's something where we're starting to look a lot more. So that's over the first 8 years of your life. And so while we were doing that study, we got contacted by an artist, Billy Apple. And so Billy Apple is a contemporary pop artist. Well, he was a pop artist. He is a contemporary of Andy Warhol. Billy is an interesting man. So he lives here in Auckland, and he contacted us and he came to see us. And so Billy, in 1970, did a display which was shown at the Serpentine Gallery in London in 1974, and it was called Excretory Wipings. And so this is a picture here of Billy in 1974 as a young man, and he effectively, for a year, he collected his first nose blow for a day and his first wipe every day. He also collected his another body specimen every day for a year as well, and that was the reason that they shut down this exhibit. So they actually shut it down and censored this, which has led to a whole lot of things about actually censorship in art, and is very interesting. But Billy, of course, being the artist that he is, when they shut it down, he just basically archived it. And so he kept it in his warehouse, in plastic, in boxes, very well done and very well kept. And he turned up and he said, well, look, I've got these samples. Can you do anything with them? And so those samples were from the 1970s. Now, he wouldn't let us sample them all because it's a valuable art exhibit, but he did let us sample some. And so what we did was we took 3 samples from 1970 and we sampled those, and Thelini did this. She sampled the samples, and she did a straightforward analysis comparing them to the human microbiome samples subjects from 2012. And so this is just a straight 16 Samplicon sequencing analysis. But you see here, this is Billy's 3 samples here from 1970, and they just sit up here with the normal ones from the Human Microbiome Project. So really, really cool. So in 1970, you would argue that Billy was actually a pretty healthy man, which is good. But of course, Billy in 2016 was a different beast. And so in 2016, Billy, of course, is an old man. He's about 80, I think he's 86 or 87 now, something like that. Although the brand is only 50 something, because Billy Apple is not his real name. It was the brand that he took. So he says he's only 50, but that's okay. But what he did anyway, in 2016, his samples, you see, are way down here in the corner. Clearly not clustering with the bulk of the people from the Human Microbiome Project, and they're showing differences. And of course, Billy's gut now is not what it used to be. He has some diverticulitis and other bits and pieces going on there. He subsequently had his gallbladder out. We have done the microbiome post his gallbladder as well. Yeah, he keeps coming in and giving us samples, so it's kind of cool. But yeah. So the question though is, what happened to his microbiome over that period of his life, right? And so previous studies have looked at microbial population structures and shown them to be stable over a period of time. And so Faith in that did it, I think, out to 5 or 6 years of age. Well, sorry, not 6 years of age, but over a period of 6 years, and with a number of people. And they showed a really nice curve in terms of Jacquard, showing that, you know, you retain part of your population, and that it drops down, but you know, over a 6-year period, sort of anywhere up to 60% of your population is still similar to what it was originally. You retain those individuals. They did a great extrapolation of this, and they extrapolated their curve right out to sort of 50 odd years, which is kind of cool. And they had, of course, their confidence interval. So we happen to have samples now that were 40-something years apart. And so when we plotted Billy and the Jacquard for Billy, it was about 48% identity with what he had originally. So over this period of his life, 46 years or 47 years, whatever it was, 48% of the organisms that he had was the same, at least, strain, or species, sorry, not strain, because we didn't go strain level, but they were the same species as what he had when he was younger. And beautifully, this, you see, this is fitting right within the confidence interval of this curve. So it's really, really nice, and indicates that over that period of life, there is a core part of your microbiome that you retain, even right through into old age, as Billy is now. And so that was really, really very good. But the other thing, of course, that we all think is that the microbiome is affected by host genetics. And so, of course, there's been a nice study that Bonder and that did, and they were linking different genetic variants within the host genome with the presence or absence of particular bacterial phyla or species and things. And so Billy, being the artist that he is, also sequenced his genome. And so we had a full sequence, genome sequence for Billy Apple. It's online, you can get it if you want, as are some of his cells. You can actually buy his cells from the ATCC if you want to. He's immortalized some of his cells. And so they're there. But Billy, of course, then we were able to look at some of these predictions from the Bonder paper. And what we saw with Billy, and you've got his SNP detail here. So the reference allele is here, the alternate allele. And there were a few that we didn't detect, which of course is part of the way that goes with genome sequencing. But for these ones, we saw pretty good ideas about what they were. So here, for example, Billy's homozygous for the G. The argument is that from Bonder was that the methanobacteria, if you have this RS4548017, I don't know where that is in the genome, but if you have that particular variant, then if it's a G at that position, then you should have low levels of methanobacteria. Now, methanobacteria were not detected in Billy in either 1970 or 2016, which is pretty consistent with that. Here, what he had is a 1023 for the reference alternate for the dialyster invis cis. And this one kind of went against the pattern though, and it wasn't detected in Billy, but whereas they thought if you had the A allele, then they should be up. And so this pattern goes through here for various other ones. Again, with the lactospiraceae, with the AG, is A's reference and G's the alternate. Billy is G, and of course it's down and he's non-detectable. So it's an interesting thing. Needs to be done in many more people. You know, this is an N of 1 study, so it's kind of interesting, but it is an N of 1 study. But there are, anyway, from that study, there are indications that, in fact, the microbiome lasts a long time, that there are at least components of it that are lasting out through sort of, 40-odd years of your life, through adulthood. And moreover, that some of that can be sort of matched up with some genetic sort of things or indications that that might work. But if the population is stable like that, and if it's stable for the preterms as well, like it's coming towards a stable population that we saw with the terms, similar to the terms, then is it really imposed on us? Is it selected? I'm going to skip this one. This is about sheep. But can we stably change the microbiome? And so this is a study that we're just finishing now. And so in this study, what we were trying to do was to look for an ability to change the microbiome by using FMT in individuals. So this is our gut bugs trial. Again, it's led by me and Wayne, and Karen Leong here done a huge amount of this work. So the idea here is that ultimately we should be able to get to a situation where we can sell basically a healthy bacterial population that people have as part of their prebiotic or probiotic diets. But of course, taking a fecal material and getting people to use it is not very appetizing. You can't just give it to people. You can't make it into a smoothie and have them drink it. That just isn't nice. So we encapsulated the microbiome in this particular study, and then we fed it to individuals. What we did is we took 9 healthy donors, and these donors were very, very strictly screened. That included looking for antibiotic-resistant organisms, etc. We encapsulated the microbiome, and we kept it for a short period of time. It was only in the freezer for a month and a half maximum. We then fed the microbiome, or in a randomized controlled trial, double blind, to 87 individuals, adolescents who had severe obesity. The individuals who were given the microbiome were not treated with antibiotics. They were given a pica prep, which basically flushed out their gut, and that was it. And that was the only treatment they were given. What we did a large number of assessments to make sure that they were eligible. So they were then, the individuals were brought in. They had a lot of criteria. I'll show you those in a second. But they were assessed for their eligibility. They were randomized either into the transfer group, and there was 20 males, 20 females, or into the control group, again, 20/20, and we did a few extras. They went through baseline assessments, which included DEXs, 6-week assessments, again, 12-week and 26 weeks, and then the data was analyzed. The donors were very strictly checked, so they were basically between 18 and 28 years of age. Their body mass indexes were between 18 and 30 kgs per meter squared. And body fat, total body fat, was less than 29% for females and 90% for males. They had to do regular exercise, regular bowel habits, intake of fruit and veggies every day, that sort of thing. They were excluded for a huge number of things. I won't go through this, but any evidence of any disorders, bowel disorders or anything like that, type 2 diabetes, anything like that. The recipients, they were aged 14 to 18. They had a body mass index of more than 30 kilograms per meter squared, and they were all post-pubertal. And they were excluded for a number of reasons as well. So these were morbidly obese individuals. Some of the boys that we had recruited into the study weighed in excess of 220 kilograms. And they're sort of 16 years of age, so they're big people. Alpha diversity levels in these individuals were the same, and so here we've got donors and recipients, and the donors and recipients, donors in green, recipients in orange, they were the same for females and males, roughly. They didn't differ between the donors and recipients across the board before FMT. After FMT, there was a shift, and so what you see here, these are the donors spreads here. These are the baseline data for the female FMTs here, so spread out. But what you see is a clear shift upwards in terms of Shannon's diversity index following the FMT. So the microbiome structure altered. Here, in terms of the placebo, there was a small shift, but it wasn't significant as opposed to the one with the FMT. For the males, the pattern was slightly different. So the males, we had very good diversity for the donors as well. But what you see is that even the healthy donors, some of them have quite low diversity, which is similar to the extremes of the ones that we saw with the obese individuals. But here you see the males, there was a shift upwards following FMT, and so an increase in the Shannon's diversity, but that was not significant for the males, and it's kind of an interesting thing. The FMT recipients, though, if we're looking at Bray Curtis again here, you do see a much bigger shift in terms of the FMT from baseline at 6 weeks than you do for placebo. So this is the difference between baseline and 6 weeks, and it's a Bray Curtis dissimilarity index. So for the placebos, you're seeing here that they had a bit of a shift, but the FMTs for females and for males both showed a much bigger shift. And then for all, here you see that as well. So showing, in fact, that the FMT works. The FMT we gave them was one treatment, basically, of 32, well, it was two treatments. It was 16 capsules on 16 capsules the first day, fasting, and then the next day they came in and they had another 16 capsules. But that was the only treatment over this period of time. And these assessments were done at 6 weeks post to that. The FMT groups showed a bigger change, and this is a total, but the Bray Curtis dissimilarity index again, and this is 6 weeks, 12 weeks, and 28 weeks. So it stayed. In other words, the shift was such that what actually happened was that we promoted, in the way we did this, actual engraftment and augmentation of the microbiome population, as opposed to a simple burst shift. So there was a stable change effectively in the microbiome over the 26-week period for these individuals, which was kind of cool. All right. The female recipients did change differently to the males. I'll wrap this up in just a minute and take some questions, but the females did change differently. And so we had a number of donors. We set up effectively a competition experiment when we did this. We didn't just use one donor, we used 4 donors for each individual, and those 4 donors were the same for the females. And the males, there was a subset, a small group, so a couple of donors differed for some of the males, but largely they used the same set. And what we actually saw there was that there was competition between those donors. So in the females, this particularly came through. So this is the female's FMT here and the female placebo. And this is the beta diversity shift towards donor profiles. And what we had here in green is donor 16 for the females, and donor 16 was what we considered to be a super donor. And so most of the female profiles who had the FMT shifted towards this donor. And as you see here, any shift here was random for the placebo. But definitely for the FMT, there was a strong shift towards the donor 16 profile. And this is really interesting. So I'll skip that one. And it's not simply down to the fact that it's the donor's diversity. Donor 16 was not the most diverse female by Shannon's diversity index that we had. Donor 17 was, in fact, that. For the males, donor 8 was the most diverse one. And there was a shift more closely. We've got donor 8. Yeah, that doesn't come through here for donor 8 either for the males. But what we did see, of course, is because here again we used full metagenomics. So we've done the metagenome and the metatranscriptome. And this time the metagenome and metatranscriptome actually worked very well. I'm not going to talk about that data, but the metagenome data gave us an ability to actually go to a strain-specific analysis. And so because we had characterized samples for the donors, etc., we can actually follow strain transfer at the level of the strain. And then that's really cool because you see instances where strains go into a population and they're stably retained by the recipients. And you see instances where they go in and then they disappear. You see over the later time points. You also see instances where there's competition between an existing species and the incoming species, but the recipient strain wins out or the donor strain wins out. And it's very, very nice, and you see that through the population. But we definitely saw strain transfers, and there were numbers of strains that were actually done. And so here, this is the baseline for the FMT population. And so they do share some strains with the donors at baseline, but those numbers increase significantly following FMT at 6 weeks, as you would expect. And what we saw here coming through very strongly, this is donor 16 for the females, and she's presenting and providing the greatest number of these strains within that FMT population or recipients. By contrast to placebos, they don't shift, they stay the same between the baseline and 6 weeks. So the numbers of strains that they share with the donors are effectively the same. So that's a background level, if you will. Again, what we see here, these are the males, and we did see strain-specific transfer for the males. And one of the donors, which is donor 8, who happened to have the greatest diversity, did seem to provide more of these strains to that population as well. And so that's really cool. In terms of that, I mean, the reason that the FMT trial was done was, of course, to try and treat obesity. And the primary outcome was a shift in obesity. Unfortunately, well, the primary outcome wasn't met, so there was no shift in obesity as a result of the treatment with the FMT. But there have been other changes in the individuals that are of beneficial to their overall health, including a shift in metabolic scores for a number of individuals, which is, we're still trying to understand that, but there seems to have been quite a significant impact on metabolic syndrome, at least for individuals who had it to start with at the beginning of the trial. So both the environment and the host impact on the microbiome both the environment and the host impact on the microbiome, whose composition actually seems to be stable for a very long time. And that's really where we've come to. There are some significant changes. You can modify the microbiome very clearly. And when you do actually do those FMT trials or studies like that, there are significant competition things that are going on. It's really amazing thing to look at. So this is my group and funding and collaborators and things as well. And so, yeah, if anyone does have any more questions, I'm very happy to take those. Thanks.
Really nice presentation. Thank you so much, Justin. Is there anybody has a question, guys?
Go ahead and type them in if you're averse to speaking on mic. It may take a few seconds for people to type if they're not. So I know there's quite a bit of art and microbiome work happening in Australia as well. Yeah. And I'm wondering, just in general, whether New Zealand is progressing on that front, I guess I would say. Are there many more artists jumping into that?
No, not that I'm not aware of, no. I mean, Billy's, he has done some works, he's exhibiting them around the world. He still does that. He was developing something with Google, but I don't know whether or not that's going forward now. Other people, no, I haven't heard too much from other artists around the country, no.
One of the reasons I ask is we have a number of artists in our microbiome center. One of them is, interested in transplants in particular, and so it'd be kind of interesting to get that group. If there were a group, that would be interesting to get that group together.
Yeah. So I mean, Billy, he considers that the artist is the artwork, and so that's why he's been doing this. He is the artwork, so he puts everything out there as medical stuff and all sorts of things. It's actually quite an interesting. It's an interesting data set you'd like to look at, and a whole lot of it is available. There's genomes available and stuff as well.
I wonder if he would be interested in making a presentation to us. That could be fun.
Oh yeah, he would. Yeah, probably. I can ask him. Yeah, there's no problem.
Yeah,
Yeah.
send us his contact. There's a comment from Erica Ganda. What, you want to ask your question, Erica? She's looking for
Sorry,
it.
I was just unmuting myself. No, it's not like really a question, and I'm sorry if I missed it. It's really interesting how you had that strong sex effect and only your females responded to the therapy. Do you have any theories on why that happened?
No, that's a really good question. So we thought maybe it was down to the diversity of the donors, but it doesn't seem to be that at all. So currently, no, we don't really have an idea why. Yeah, we
Good
don't.
news for me.
Okay. Yeah. No, we haven't worked that out yet at all. No. Are you doing something similar?
No, but I'm female, so I can use it. You can.
Yeah, it did make a difference in the males. In the females, the FMT actually shifted the ratio of android gynoid fat. So whilst it was a significant, that was a secondary outcome, and that was significant as a result of the FMT. So the ratios of AG fat shifted in the females. Somebody asked me a question, are we going to look for microRNAs and microtranscriptome data? Yeah, so we will. Yeah. We haven't yet, but we will. Oh yeah, no, we didn't look at Billy's nasal microbiome, and we should. I haven't broached that with him, but we could go back and do that. Yeah. He would just give us the samples, I think, but we haven't done that one, no. Yeah. Cool. Well,
But just
yeah.
last question. The fish population that you logged to again, were you able to identify if they are lytic phage or prophages in the sequence of the bacterial genome, or it was just lytic phages free particles, or you didn't study that?
So that was all derived from the metagenome, so it's all DNA sequence, but we didn't look to see specifically whether they were integrated into the genomes or they weren't. Arguably, being the DNA, it should contain the lysogenics, probably more of those than the lytics, but that wasn't confirmed. The studies that Tommy's going on to do now, he's looking for separation of the viral particle or the phage particles from the samples, and he's going to look at both the RNA and DNA components. So in the other studies, we have both the RNA and DNA components, and so from that we can then look at different aspects of that question. But in this one, no, we couldn't do that, unfortunately. But it's a good question. It's definitely something that we have to look at. And it's really cool. I mean, they're amazing things, right? It's but no, we just don't know. I don't know. Someone says, what would it look like with trans individuals? I have absolutely no idea. I mean, maybe it is hormone related. It could be something like that, but the difference for female male, that's an obvious place to look, but I don't know that anyone's ever done a study on FMT study on that.
Well, I was going to say, I'm especially curious within trans individuals, because we ascribe it to a genetic effect, but how much of it is genetic versus hormonal, and you just don't see that difference because we always see on either extreme.
Yeah, I have absolutely no idea. Yeah.
Oh, we can't hear you clearly if you're speaking.
Oh, sorry, I have no idea. Yeah, yeah, I hadn't considered that question. So, yeah, I don't know.
Okay, and